Good morning, everyone. Is there any question, any doubt so far? So, all right. Yesterday we have seen how to generate the alignment file. So, for each sample, we got two files: summary file and the BAM file. So, summary file will tell you how much percentage of the alignment is there. So, you can see total number of reads, and these many numbers are reads are unpaired. Why this is showing unpaired? Because this is single end data. If it is paired end data, it will tell you how many paired reads are there and how many reads are mapped properly paired, align exactly one time and more than one time, and total percentage. So this is individual summary files. If you would like to consolidate one report all for all the samples, we can again use the multi QC. So from here, this time we have to select the high set 2, and then we will select the mapping summary files. And then execute. Then this will give us the consolidated graphical report from all the samples for the alignment summary. So this will just tell you whatever in the summary file is there, nothing more than that. If you would like to know individual samples, individual chromosome wise alignment, then you can use QualiMap. So QualiMap is a tool you can use for any BAM QC generated from the alignment, not from the de novo assembly. So QualiMap BAM QC, we will see why this is happening, but this is the correct way to do. So we will do the QualiMap BAM QC. We will select the BAM files. Reference genome, if you have some regions, you can give one file here, otherwise we will give the whole genome. So if you want per base coverage output also, you can select yes. Otherwise, just execute this. Sorry, I deleted the wrong files actually. Anyways, I will just execute again. I have to delete this QualiMap BAM QC. Select all the BAM files. And then run this. Let's see why multi QC did not run. This is for the alignment, not for the Trimblur we used for the raw data, not for the alignment. So this is the QC for the alignment, not for raw data. So meanwhile, this is running. We will see how we can visualize the BAM file. So for the BAM file, if we come here, this is our alignment file. So if we click here, display, then our BAM file will open in this pane. So this is the BAM file. These are the columns: query name, flag, lead name, position, mapping quality, cigar, MRM, position, insert size, sequence, and the quality for each page, and the some optional information. So this is in BAM file, this is a header. First line of the header will tell you whether your BAM file is sorted or not. And if you will come at the end of your header, you can see this is the program which we used for the alignment. So these two rows in the header are common for any organism, any BAM file, but this length of the header will depend how many chromosomes you have in that organism. This is the read name. These are the different columns. Column names are up. This is the read which is aligned to the reference, and this is the quality of each base which is aligned. So this is your BAM file. Now if you would like to visualize this BAM file graphically, so this, if you will click just below this line, header line of this BAM file, this pane will expand. And here if you will see, there are different tabs. So this is for the help. This is for if you would like to see what input you have given to generate this BAM file. This is for the visualization. This is for to rerun again. This is for to dataset detail. This is for the link, and this is to download in your local system. So we can click on the visualize button. So then these options will open here: display at UCSC main, display at IGB, display in IGB, display at BAM IO Bio. So UCSC is a web browser, BAM IO Bio is also a web browser. But IGB and IGB you have to install locally. You can install any one, both are not required. These programs are Java based programs. So before you install IGB or IGB, you have to install Java, and then you can install IGB. And before you click on these links, first you have to keep your IGB open. So I usually generally use IGB, but this is your choice. Both are good tools, IGB or IGB. So meanwhile it is opening, let us see. This is open. So this is how the IGB will look like, the main page. And if we click on the display at UCSC main, it will open in another browser. So this is the UCSC browser. Now you can see, and this is the BAM file. On the top, this is the alignment file. Say suppose if, currently it is showing for the chromosome X, you can change this position. If you wish to visualize for one particular gene, you can also keep. Say suppose I want to use PTPN, one gene, then I can just put the gene also. Then BAM will automatically show you the PTPN gene. So this is over here. So like that you can change this visualization. You can change the chromosome and the what regions you want to see. So this is right now it is selected these base pair, and you can see here these numbers are here. And this is for the chromosome 20 because PTPN1 gene is located on chromosome 20. So like that you can visualize in your browser. Coming back, if you click on the IGV local, it will open in here. And here you can see automatically human genome reference is selected. If it is not selected, you can select it from this list. So these are all the genomes which are already present within IGV. If you see your genome is not present in this particular list, you can upload. So genomes, load genome from file, load genome from URL. So both are possible. You can do all these things from here. So BAM file is over here. We can select some chromosome. So the chromosome will display at the top related to genes present on the chromosome 1 will be displayed at the bottom. And then if you will zoom in your file, and this file you can hold this pane, middle pane with mouse, and then you can move left and right. And then you can expand this. Then your alignment file will be displayed over here. So you can see how your alignment file looks like. So the solid lines are your exons, thin lines are your introns. So you can see how your reads are aligned over here. If you want to see more clear picture, you can still zoom in, and like that you can visualize. So in any, if you will increase more, then you will be able to even see the bases. So these are the bases in this gene. And on the top, this is the coverage for this particular region. Now, if you will look into this file, if any variation is there with respect to reference, that also will be able to show here. So now you can see almost in all the reads, this base is changed from G to A. So like that, you can also see what base is getting variant and where it is located. So this is the G to A variation. So once, so this might be a reason that if you will see if in the gene expression, this gene is displaying either overexpression or down regulated, then this is the variant which is responsible for modulating the gene expression. This is how you can do that. If you will come back over here, if you will click on BAM IO Bio, then it will tell you the quality of your alignment. So this is one, 2-3 tools are to visualize your BAM file in terms of your alignment and other things. This will tell you the read coverage, segment length and other things, and the alignment mapped. So all that will show over here. So this is for all the chromosome. You wish to see some chromosome, you can see. Even you can see on the chromosome 2, reads are not aligned. So you can fetch these chromosome only. So from your alignment, you can just take these chromosome because only on those chromosome your reads are aligned. So this is chromosome 2, chromosome 5, and so on. So like that, you can visualize your coverage and other parameters per chromosome. So coming back here, still it is running. This did not work. I will see, and then tomorrow we will resolve this issue. So let us run the quali map. It is done. So how it will be displaying over here. So this is the report you will get. This is the HTML report. And if you will come down, so reference size 3.2 GB, number of reads, mapped reads, secondary alignment, how many reads are alignment, overlapping read pairs, because this is a single line data, so it is showing 0. Then ATGC content, coverage, mapping quality, mismatch and indels, it will give you. That all you can see here. These are the chromosome wise statistics. And here the graphs are. So this is the chromosome wise coverage. This is the histograms. So there are different type of displays are you can use. So this is for the ATGC content. This is for the GC content. This is the clipped file. This is the homopolymer indels. So you will get different type of visualization over here. So now when you are generating this raw data for Qualimap, if you will open this, so this is the individual graphs, individual files. So now from this also you can run the Qualimap. Multi-QC to consolidate that. So here now this time you have to select the Qualimap. So Qualimap, BAM, QC or RNA-Seq output. So Qualimap, BAM, QC and then this BAM, QC report if you will give, it will show you the so still some Qualimaps are running. Once this is done, then this will start. Okay, this is done. All the Qualimaps, you can have a look at the Qualimap report for each and every sample. This is how you have to run this. So now from this alignment file, the next step is to make the count matrix. So what is count matrix? As I mentioned, count matrix is nothing but it is a counting of the reads which are aligned to each gene or the feature. So for that we will be using feature count. So this will count the reads, how many reads are aligned to each gene for each sample and give you the table. So for this we will be using the BAM files. We don't have the standard information. So gene annotation file, either locally cached, which feature counts built in or from the history. So feature count has the inbuilt annotation file for the human genome. So we will use this. So now you can see automatically it has selected HG38. Output format, so it will generate the count matrix files which is compatible for DC, HR or REMA. So these are the three packages for the differential gene expression analysis. So if you want to create a gene length file, you can create this gene length file. So these are the some options which you want, you can do it. But there is no requirement for this. Read filtering options, I don't want anything because we will do the manual filtering. Advanced option, we do not want. So execute this. So now this multi-QC, if you will look at the multi-QC from the BAM file, it will show you in this way. It is not running, but it is not giving some error. Sometimes it is giving error. I think there is a problem with multi-QC. So sometimes it gives some this kind of things. So don't worry. Then after some time you can execute again. Then it will run. Because if they are doing some maintenance and updation of some softwares, that time it will not execute your command. So then you have to take care and then try to rerun these commands in the same way. So this will tell us the feature count for each sample from the alignment files. We did the trim glore already, QC sequence and QC. Check count. In between any, so this will take a while. If you have any question, you can ask. So alright, feature counts are done. Let us have a look at it, how these files look like. So this is for one sample. So this is the count data, how it will count. So this is the gene ID. For this gene ID, how many reads are aligned for this sample. This is the summary. How many total are assigned, how many are unmapped or unassigned, unassigned read type, singleton, mapping quality, chimera, fragment length, multi-mapping, and the no feature and ambiguity. So this kind of matrices it will give you for the summary. If you want to see the feature length, so whatever the reads are aligned, it will give you the length of those features. That is the gene. So this is the gene ID and this is the length of the reads which are aligned to these gene IDs. Right? Then from this feature count, we will run the differential expression. So for differential expression, we will use the DE-seq2 package. You can use anything actually. You can use string tie, so you can use Lima, you can use Edger. So there are multiple tools which you can use. So this is the DE-seq2. So we will be using DE-seq2. So select dataset per label. So what is our factor name? So we would like to see the cell specific marker. So we will use cell specific genes. So what is our first factor? So we have two groups in our raw data. So 2021 to 22 are the tumor associated macrophage cells. 31, 32, 33 are the tumor cells. So first we will give the control treated. You can give anything actually, if you whatever the way you want. So you can calculate treated versus control, or you can calculate control versus treated. So in the control we will give the tumor cells. So first we will give the tumor cells. So this is our control. And then we will select these feature counts. So 1, 2, and 3. So we have to be careful what group count we are giving here. Right? And then we have the treated macrophage. So this is our treatment group. So for that we will select our counts. So these are the counts. So our files have header. Yes, our files have header. Choice of the input data. So this is the HTC count, feature count, or the string type. So you can use HTC also for the counting of the reads to create the count matrix. You can use string type also to make such kind of matrix. So we do not want anything to select. We want to use the default parameters. In the output, if you want any other file, so if somebody is working on the machine learning models, you can have the normalized count, and then normalized count instead of the raw data, you can use normalized count for the machine learning models. You can get the R log normalized table. This also can be used for that, and then execute. So this will give you the differential gene expression data. So alright, this is done. So if you will see the result, so this will look like that. So gene ID, base mean, log to fold change in standard error, p value and the adjacent p value. So this is for the plots. So these are the group variance within the group and between the groups. So this is the within group variance. This is between the group variance. This is the sample to sample variance. This is the dispersion for the normalized data. This is the frequency with the p values, and this is the volcano plot. You can see this is the normal fold change. This is the upregulated and this is the downregulated genes. So if you want to work with some genes, you can select these genes because this is highly expressed. So we will see how we can do the downstream analysis from our differential count file. So this is the normalized count file and this is the R log of the normalized count file. So this is how we will get the differential file. So now there are different ways to do the annotation of our DEC2 results. So we have the annotation file. If we don't have the annotation file, in the data library, go to data shared library. There is a folder called annotation, genome and annotation. So click on it and click on annotation. So here we will see Homo sapiens GTF. So this is the annotation file. Click here and then export to history as a data set. From here we can select the history. So this is our history or the project where we would like to take this annotation file and import. And if we will see, we have our GTF file over here. So this is our GTF file. So if we will see this GTF file, how the GTF file looks like. So chromosome 1, from which source it is being taken, what is this feature, whether it is a transcript or exome, start position, end position, on which strand it is, and these are the ensemble ID and other features for this particular gene. So now before we do intersect our annotation with our DEC result, we can convert this GTF file to bed file. So there is a tool called GTF. So GTF to bed. There is a tool called. So directly also you can see append annotation from GTF to differential expression tool output. So you can click over here and then output of DEC or Azure. Select here. So this is the DEC output. Input file type DEC tool Azure Lima. Reference annotation file. So this is the annotation file and execute. Then our annotation will be append over here. So this is one way of doing the annotation of the genes. So and let us finish this and then we will see how the results come and how to interpret from here. Let us have a look how this annotation file looks like. So you can see in this header is not there, but the columns are same. This is the gene ID, base mean, log to fold change and other things. So now you can see this is the annotation, but it is not annotated properly. So that's why we don't use usually annotation from here. So it will give for most of the genes, it will give NA, right? So you can download this file and have a look at this also. But you can't see anything from this file. This file you can open in here, and this is not a very good way to annotate the gene for the DSeq output. So if I go to my folder, this is the file. I will show you how this file looks like. So this can be opened in an Excel. So if I go and open this file, so from here we can select all the files, and this is the file. So you can see everywhere, if you will put a filter over here, you want to see, so you can see everywhere it is NA. So this is not a very good way of doing the annotation, because sometimes what the reference you use, what the annotation you use, that might differ. So this is not a very good way. So to do that, we will be doing the manual annotation using the differential expression file. So for that, what we are going to do, we are going to download this result. So this is how this file looks like. So we will download this file, and then this file also can be opened in an Excel. So if we open this file, so this is the file. Next, next and finish. So you can see in this header is not there. To incorporate the header, we will just insert one line, and from here we can copy the header. So this header, if we will copy, we can paste that header over here. Now this is with the header. So this is our differential gene expression file, and with this we will be doing the annotation. So towards that, first we have to filter our file. So we can see how many total genes are there which are differentially expressed. Now you can see for these genes there is no value. So for that, first we will refine our file. So how many genes are there? So they are total 28,395. So we can relabel that total 28,395. So this figure will tell us how many total genes are there. So now first we will put a filter on the p value, and from here first we will deselect everything and just select NA. Now we need to remove these values because they are not significant. So select all these rows and say control minus. So these cells will be deleted, but without having any gap. If you will just simply delete, then this will remain the gap in your file. So you remove the filter. Now this is the filtered file. Again you can check whether any NAs are there or not. So now you can see NAs are not there, they are removed. So now first we have to, so how many are there now? So after this cleanup, we are left over with 23,808 genes. So this filter you can put on adjacent p-value. If you are considering the adjusted p-value, then you can select NAs from here also. You can remove these also. Remove the filter. check how many are left over. So these are left over with 19,175. So keep this original file. Don't touch this file at this time, because if you want to do reanalysis, you can use this file. So we will just copy this file and paste it in another sheet. So this is the total P adjusted. And these, how many genes are there? So these are 19,000. We can relabel this so that we know how many gene features are left over. 19,174. So now we will select from this how many are significant. So put a filter on the p value. Select number filter less than or equal to 0.05. So this is the p value we have to keep, 0.05. So now these are the significantly different. So these are the differentially expressed genes. These are the highly significant differentially expressed genes. So how many are there? They are total 7,397. So now from this we can copy this sheet and paste in another sheet. So these are our significant differentially expressed genes, and how many are there? They are 7,397. Right? So for these genes, because these are the significantly differentially expressed genes, so this we will take for the annotation. So this all downstream analysis from the or if you want only few upregulated, downregulated, because this is the significantly expressed. So you can do another way also. Put a filter here and select log to fold change number filter, and then the greater than or equal to 2. So these are the upregulated genes. So here you can put the status, and these are our upregulated genes. And you can label them in the red color. So remove the log to fold change filter, put a filter again, number filter, this time lesser than or equal to -2. So these are the down regulated genes. Put a down regulated over here. And then this will not come like that because there are gaps in between, but what you can do, just you copy this and paste it over here and make them as a green color. Then these are our these are our down-regulated genes, and we will remove the filter. So now rest of the genes, they are normally expressed. They are nothing to do with that. So these are the upregulated and downregulated genes. Now for this, we have to do the annotation. So for today, I will stop here, and tomorrow we will see the annotations of these genes which are down-regulated and up-regulated genes. So again, how you can do, you can either take all the genes, then you will know what genes are not differentially not up-regulated or down-regulated, they are normal. But if you want to do only up-regulated, down-regulated, you can again put a filter over here, and from this you can just remove the blank and just select only up-regulated and down-regulated and copy this. So now total how many are there? These are total 3973 genes which are either overexpressed or down-expressed. So this particular sheet again you can copy and then copy it in this new sheet. So then this is your significant up and down regulated genes and how many are them. So you can see these are 3973. So 3973 genes. So these are the upregulated and downregulated genes. If you want to separate how many are upregulated and downregulated, select this column, insert recommended chart, and just select the column chart. Or you can insert the pivot table. And then maybe in this worksheet and select this column here. Because this is we have just copied, so copy this and paste special only the values. Right? And go to file, first save as, browse, and then I will save it over here in the Excel workbook, and I will give this name RNA-Seq only. Now this is here. You can put a status. You can make the count of here. So you can see these many genes are down regulated and these many genes are up regulated. And then you can create the chart out of it. Copy this, paste it over here, and then insert recommended chart or whatever the chart you want, whether it's a bar chart or a column chart, anything, you can do that. And then you can paste it over here. You can delete these bars. This, and then you can label this. You can change the view however you want. Add chart elements, data labels. You can see outside bar, so either in the number or you can change these numbers. So you can put this trend line. If you want to draw the trend line, that also is, you can see legends. You can do that. Data label, go to more labels, more options. So instead of the number, you can change the options from here. So there are many things which can be done from these charts. So this is how you will see how many are up-regulated and how many are down-regulated. So the same thing you can plot it from here. So then you will be able to see even the normal gene counts, how many are there. So there are many things which can be done from this data. So tomorrow we will be seeing more analysis from this data, how to do annotation, how to do all these CAC pathway analysis, gene ontology, how to create all these visualizations, and how to plot the gene to gene interactions between these genes, what we are getting the significantly differentially expressed. So till here, if any question is there, please let me know. So is there any question?
Hi, can you hear me?
Yes.
Regarding P and P adjusted, what is the basic difference?
So basically, I am also not very sure about this, but when you calculate the p values, there are some errors. Okay, so how you calculate those errors? Because when we calculate anything, there will be some probability of errors. So when we calculate the p value here, there will be some errors. So computationally, it will calculate those errors and then adjust those p values. So then it will calculate your adjusted p values. So this is after the error correction, that p-adjusted value, that is after error correction. So now earlier the concept was to consider the p-value, but over the period of time, as the technology, as the algorithm develops and they improve, so then now people are considering the adjusted p-value after calculating the errors.
Okay. So is it always like P or P adjusted should be adjusted with 0.05 or is there a range?
So there are two concepts to say significant that you can choose, but there are only two levels, whether at the 95% level, so that is 0.05, or the 90% level, that is 0.01. So as you increase your significant level, then you are more stringent. So you have to give some relaxation. That depends on which biological system you are dealing with, looking at the variability as well as what type of analysis you want. Say, suppose I want to call for some variants which is related to my patients. So there I need more more specific, more sensitivity. So I just these p values at like 99% level. So this is how it goes. But in RNA expression, we need to be a bit relaxed. But this is guidelines, only two values, 95% and the 99%. So that is the confidence level of your significance.
Okay. And when you're comparing two experimental sets, we have p-value as well as we have log to fold change.
Yes.
So what is the best way to differentiate between two sets? We use based on log to fold change.
So how you can say that your log to fold change is significant or not? So to say that yes, your differences are significant, so that is how p value does matter.
I did not get your point.
Say suppose you have the fold change, right?
Fold change should be 2, right?
Yes. So when you want to compare the fold change, right? So you have one value for one data, you have one value of another sample, right? You can get the differences, but how you will say that this difference is significant or not?
For that, we use
How
p.
Yes, that is how you have to calculate your p values. That will tell you whether that change is significant or not. Say I say that okay, I am doing one value is 3, another value is 2. Okay. Another set of the values I have 20 and 18. Okay. Both are 2. Both are like the differences are 2, but whether they are significant or not. So look, values itself, I can say that yes, that change is not significant, right? Because I am getting the difference of 2, whether 20 or 18, right? It is not significant, but you cannot just looking at this, that difference is, that change is significant or not. So p value will tell you whether that change is significant or not.
Okay. So suppose I have a file which is already processed, and further I'm going to see which is the most upregulated and the most downregulated, like which comes in the top 50 and the bottom 50. Say, I want to see which is the most affected genes out of all the thousands of data sets.
This file is there. So what you can do is come back over here or maybe here also. So put a lock to fold change filter. Then select this and go to this time you go to number filter. There is an option called top 10. So you want, say suppose you want 25 top genes, go to top 25. So these are top 25 upregulated genes. So
Okay, so that is based on lock to full change, right?
yes,
Top,
yes,
top.
yes.
Okay.
So then, if you want, so this is upregulated. If you want to see downregulated, clear this filter, come back here again, number filter, go to top. From this, you can go to bottom.
Okay.
So these are the downregulated top 25 genes. So this is. So this all we will see tomorrow.
Okay, okay. Thank you so much.
So any other question? So if no more questions are there, I will stop it over here and we will meet tomorrow. Thank you so much. See you tomorrow.